Alright, good morning all of you. So, very nice to talk to you all again. So, last session we did a beautiful topic, the histology of the liver and the pancreas. We analyzed some very nice logic. Today we go into a really important topic, the process of digestion in the body. How does food actually get digested with the help of different enzymes? So let's analyze the physiology of digestion. सबसे पहले हमने माउथ के अंदर फूड डाला, so how will digestion take place in the buccal cavity? How will it take place in the mouth? Mastication, which is chewing of food with your teeth, will break down the food into smaller particles, and tongue mixes the food with saliva secreted from the salivary gland. So tongue converts that masticated food into a rounded ball which is called as a bolus, and tongue will help the bolus formation with saliva. Salivary glands will give out saliva. Saliva has two important enzymes, namely ptyalin and lysozymes. Ptyalin is also called as salivary amylase. याद है मैंने आपको सिखाया था when we had talked about parotid gland, submandibular gland, sublingual gland, that there is secretion of saliva which has in it ptyalin, also called as salivary amylase, and salivary amylase or ptyalin acts on polysaccharide starch and converts it into a disaccharide maltose. So what is a polysaccharide? शुगर का बेसिक यूनिट होता है ग्लूकोज. So glucose attached to another glucose is a disaccharide. Another glucose, another glucose, another glucose, another glucose. A chain of glucoses is called as a polysaccharide. And starch is a polysaccharide. So suppose we eat rice, or we eat potato, or we eat bread. All these are rich in starch, which are polysaccharides. And salivary amylase or ptyalin breaks down the starch, which is a polysaccharide, into maltose, which is a disaccharide. So what does starch polysaccharide break down into? Glucose and glucose, maltose. Glucose and glucose, maltose. Glucose and glucose, maltose. So a polysaccharide now becomes a disaccharide, which is called as maltose. And this happens in the mouth at a pH of 6.8. So saliva is slightly acidic, and at a pH of 6.8, polysaccharide starch becomes disaccharide maltose. And by this mechanism, 30% of the starch is hydrolyzed. 30% of whatever starch we have eaten is broken down. Breaking in the presence of water is called as hydrolysis. To break is called lysis. In the presence of water is called hydrolysis. And 30% of the starch is hydrolyzed into maltose at a pH of 6.8. That happens in your mouth. And saliva also has in it lysozymes. Lysozymes are antibacterial and they prevent infection. Lysozymes will be antibacterial chemicals. They kill the bacteria present in the food. Do you all remember I have mentioned the word lysozyme? In the intestine were found Paneth cells, and Pannate cells also produce lysozymes which kill the bacteria. So, pannate cells in the intestine and lysozymes in the saliva both are antibacterial and they prevent infection. And the bolus form then is pushed into the esophagus, and that process is called as swallowing, also called as deglutition. So swallowing or deglutition takes place. And food is swallowed too quickly for all the starch to be reduced into maltose. Tyline is swallowed in the swallowed food continues its action for 15 to 20 minutes in the stomach before it is inactivated by the acid of the stomach. So sir, tyline would have broken down all the starch, but how much is actually broken down? 30%, because within 15 to 20 minutes the food has passed through the esophagus by peristalsis and food enters the stomach. And in the stomach, do we remember, there is HCl, and hydrochloric acid will break down tyline. So tyline टूट जाएगा by the action of hydrochloric acid. So how much of the starch gets digested? About 30%. So tyline reduces 30% of the starch into disaccharide maltose. Now, from the buccal cavity, food will pass through the esophagus and enter into the stomach. Stomach has in it gastric juice. Gastric juice is made up of HCl, pepsinogen and mucus. HCl, pepsinogen and mucus, I hope you remember, HCl given by the parietal cells or oxyntic cells, pepsinogen given by chief cells or peptic cells, and mucus given by mucus cells. What will they do? HCl stops the action of salivary amylase. As I just said, HCl destroys salivary amylase and only 30% of the starch gets digested. HCl kills the germs that may enter along with the food. Stomach में HCl होता है, the pH of the stomach is 1.8. Very acidic. It will kill the microorganisms which are found in the food. And HCl helps to soften the food. It will make the food become softer. And HCl provides an acidic medium pH of 1.8, and that HCl at a pH of 1.8 converts pepsinogen into pepsin. If you remember, what are your chief cells giving out? Pepsinogen. And pepsinogen is an inactive chemical. It becomes active chemical called as pepsin by the action of hydrochloric acid. So HCl converts pepsinogen into active pepsin, and pepsin acts on the proteins in the stomach. Pepsin acts on proteins and it breaks down proteins into smaller chains which are called as peptones and proteoses. So, pepsin breaks down protein. What is a protein? A chain of amino acids. तो अमाइनो एसिड्स की बहुत बड़ी चेन को प्रोटीन कहते हैं. पेप्सिन ब्रेक्स द बिग चेन ऑफ प्रोटीन इनटू स्मॉलर चेन्स व्हिच आर कॉल्ड एज प्रोटियोसेस एंड पेप्टोन्स. So where does digestion of carbohydrate begin? In the mouth by the action of salivary amylase. Where does digestion of protein begin? In the stomach with the help of pepsin, which was made from pepsinogen under action of HCl. What does pepsin break down protein into smaller pieces called as proteoses and peptones? And we have mucus. Mucus has in it bicarbonates, and bicarbonates are alkaline in nature, and mucus with bicarbonates forms a protective layer on the mucosa of the stomach and prevents the action of HCl. अभी stomach इतना acidic है, 1.8 pH है, the HCl would destroy the internal lining of the stomach, which we remember is simple columnar epithelium. तो HCl उस columnar epithelium को destroy कर देता था, who protects the gastric mucosa and the columnar epithelium from the very powerful hydrochloric acid mucus. Mucus forms a nice coat over the gastric epithelial cells, and it is rich in bicarbonate, full of alkaline chemicals, and mucus, which is alkaline, neutralizes the effect of hydrochloric acid and protects the gastric epithelium from the action of HCl. From the stomach, we have a nice logic, renin. So, renin is the proteolytic enzyme found in the gastric juice of infants, which helps in the digestion of milk protein, and small amount of lipases are also secreted by gastric gland. सर आपके स्टमक में लाइपेज सिक्रीट किया जाता है. लाइपेज नाम से मालूम पड़ रहा है should digest lipids, but HCl will break down lipase. So lipase secrete तो होता है, but HCl lipase को तोड़ डालता है, इसलिए lipids cannot get digested in the stomach. So HCl breaks down lipase and prevents the breakdown of fats or lipids in the stomach. And in the gastric juice of infants who are less than 1 year of age. Sir, 1 साल के नीचे तो बच्चा सिर्फ मम्मी मिल्क पीता है, mother's milk is being fed, and the stomach of an infant, nature has given it a special enzyme called as renin, and renin found in the gastric juice of infants helps in the digestion of milk protein. So which milk protein? If you have heard of it, milk has in it a protein called as casein, and renin will break down protein called as casein into para-caseinate, and para-caseinate बन जाता है. Para-caseinate combines with calcium to form calcium para-caseinate, and calcium para-caseinate is very easy to get digested in the stomach of the child. So renin given by the stomach of an infant will break down casein into para-caseinate, combines with calcium to form calcium para-caseinate, which will be easier to digest for the child. So ये होता है renin, and once digestion takes place in the stomach, sir, secretion of gastric juice होता है. It will churn the food for about 4 to 5 hours. 4 से 5 घंटे तक churning होता है, and the solid food now becomes a yellowish color, semi-solid paste-like fluid, which is called as chyme. So we had eaten, that was food. What the tongue did, deglutition, was a bolus. And after stomach has done the churning, it has now become chyme. Chyme acidic in nature. And chyme will further move into the duodenum by opening of the pyloric sphincter. So I hope you remember, pyloric sphincter will open and stomach contracts and chyme passes into the duodenum. It takes 45 minutes for the stomach to become empty, which is called as gastric emptying. So churning is for 4 to 5 hours and gastric emptying is for about 45 minutes. All the acidic chyme now enters into the duodenum. What will happen now? Sir, food enters the duodenum, which is the uppermost part of the small intestine. Now, this food in the duodenum called chyme is acted upon by liver, which produces bile, pancreas, which produces pancreatic juice, and intestine, which produces intestinal juice. I hope you remember, mucus entericus. So, bile from the liver, pancreatic juice from the pancreas, and intestinal juice or mucus entericus from the intestine all act on the food in the duodenum called as chyme. So, सबसे पहले let's talk about bile. Yeah. So, bile helps to neutralize the acidic chyme. Bile is alkaline in nature. It will neutralize the acidic chyme. I hope you remember, in the duodenum, in the submucosa are found Brunner's gland. And Brunner's gland will neutralize the HCl of the stomach, because Brunner's gland has in it a lot of mucus. And also bile produced by the liver will help in neutralizing the acidic chyme. And bile juice is a yellowish green color liquid. Bile juice is yellowish and green. And it is made up of water, cholesterol, which is fat, bile salts and bile pigments, and phospholipid, but no enzyme. Sir, what are enzymes? Enzymes are chemically proteins. So, mouth had in it salivary amylase, which is an enzyme, protein. Stomach has in it pepsinogen, pepsin, renin, which are proteins. Intestinal juice, pancreatic juice are again proteins. But bile, और ये खास याद रखना, bile is made up of water and cholesterol, fat, but it does not have enzymes. There are no proteins found in bile, and it has bile salts and bile pigments. And what does they do, sir? Sir, सबसे पहले you have bile salts. Bile salts are given in the bile. Bile salts, you may remember these names, and also please note it down in your book. आपके books में ये दिया नहीं है. Sodium glycopholate and sodium taurofolate are the bile salts. Which are the bile salts? Sodium glycopholate and sodium taurofolate. Sodium glycopholate and sodium taurofolate will help in the emulsification of fats, in the physical breakdown of fats. Physical breakdown is called as emulsification. Mind you, bile salts don't help in digestion of fats. Fats don't break down into simple substances, but fat के बड़े-बड़े globules are broken into smaller pieces, and that is called as emulsification. And those smaller pieces are called as micelles. What are the small pieces called as? Micelles. So we have micelles, which are breakdown products of emulsification. So bile salts, sodium glycopholate and sodium taurofolate physically break down large fat molecules into smaller droplets, which are called as micelles. And bile salts also activate lipases. Sir, पैंक्रियाज में से लाइपेज आता है और बाइल सॉल्ट उन लाइपेजेस को एक्टिवेट करते हैं, एंड लाइपेजेस फ्रॉम द पैंक्रियाटिक जूस will help in actual digestion of fats, which are now called as micelles. So who will actually break down micelles? Micelles are actually broken down into simple substances by pancreatic lipase. And who activates the pancreatic lipase? Bile salts, sodium glycopholate and sodium taurofolate activate the lipase. So activation of lipase is done by bile salts. And also we have bile pigments. Bilirubin and biliverdin are produced as a result of breakdown of hemoglobin. I hope you remember, in the liver are found Kupffer cells, and Kupffer cells digest old RBC, and then RBC will break down, उसमें से hemoglobin निकलता है. Hemoglobin is further broken down into heme, which is iron, and globin, which is a protein, of which heme or iron is transported from the liver into your bone marrow, और bone marrow में iron is again used to make new hemoglobin, and hemoglobin में से हीम तो निकल के bone marrow में जाता है, पीछे बचा globin. That globin protein in the liver is first converted into a pigment which is called as biliverdin, and biliverdin is later converted into a bile pigment called as bilirubin. So once again, what does Kupffer cell break down? RBC. After breaking of RBC, what is produced? Hemoglobin. What does hemoglobin break down to form? Heme and globin. What happens to globin? It is broken down first into biliverdin, and biliverdin then becomes bilirubin, and bilirubin then goes from the liver into your blood, and via blood enters the kidney, और kidney में bilirubin gets converted into a chemical called as urobilin, and urine में urobilin पास होता है. It gives your urine a pale yellow color. तो यूरिन में यूरिया, यूरिक एसिड, ये सब मैंने बताया था पास होता है, उससे यूरिन में कलर नहीं आता है. Where does the color come from? Urine color comes from urobilin. Where is urobilin produced from? From the pigment called as bilirubin. Where is bilirubin produced from? Biliverdin. Where does biliverdin come from? Breakdown of hemoglobin, and from globin is made biliverdin, from it bilirubin, from it the urobilin, and urobilin gives your urine a pale yellow color. Some of the bilirubin also goes into your stools. तो लिवर से थोड़ा बिलिरुबिन ब्लड से किडनीज में जाता है और थोड़ा बिलिरुबिन passes by bile duct into duodenum और duodenum में bile pigment bilirubin and biliverdin pass through the intestine and get converted into a chemical called as stercobilin, and stercobilin gives your stool the dark brown color. तो stercobilin से आपका stools का color आ जाता है. So who gives urine its yellow color? Urobilin. Who gives stool its color? Stercobilin. What are these chemicals made from? Biliverdin and bilirubin. What are biliverdin and bilirubin called as? Bile pigments. So that is bile. And next we have pancreatic juice and intestinal juice. But yes, we'll just take a small break over here and we'll go into the final step of digestion, pancreatic juice and intestinal juice. See you.